Okay, now let's look at specific defenses, adaptive immunity. Sometimes this is what's called just plain immunity, the other being just defenses. So it's the third line of defense. When this kicks in, which takes a little while, it doesn't mean the other things stop working. It just means this is added as in addition. And so this is the response of the body against very specific pathogens or some product of the pathogen, like a toxin or something like that. Lymphocytes and macrophages and the phagocytes are the cells that are most involved. The phagocytes are not specific, but the lymphocytes are. There's a couple of things. One is that this system is specific. It is very specific for each specific antigen and for each specific pathogen. And it has memory. So once you encounter that pathogen once, your response to it will be stronger and better than the first time that you encounter it. And this is the basis for vaccination and why you don't get the childhood diseases later in your life. So if we look at this, the first line of defense, keep the buggers out, build the wall. Then second line of defense are phagocytes and natural killer cells, inflammation, fever, and interferon and complement. The third line of defense is the specifics, cellular immune response and the humoral immune response. So we'll talk about each of those separately. But both of these responses are about the antigen. And the point of the specific responses and the specific immunity is to eliminate the antigen. That's the bottom line, is getting rid of the antigen. Now, the antigen is that aspect of the pathogen that elicits an immune response, that makes the immune system happen. I often in class will use the analogy of what are the characteristics of a person that you're attracted to? Like, so, girls, do you like tall guys or do you like shorter guys? Like, what is the aspect? So that would be, that evokes a response. Do you like dark complexion? Do you like fair complexion? Do you like blue eyes? Do you like brown eyes? Do you like muscular, athletic bodies? Do you like more studious, bookwormy looks? Do you like him to have a good job and lots of money, or does that not matter? Do you care about how he's dressed? Do you like fashionable shoes? Or whatever the aspects are that will evoke a physiological response within you are different aspects of that person. The antigens are the different aspects of the pathogen that will evoke a physiological response in the immune system. The more boxes they get ticked, like the more antigens there are, different antigens, then the stronger the response against that pathogen will be. So the antigens can be proteins, they can be polysaccharides, but they tend to be organic large molecules found on the surface of the pathogen. Now, all cells have got these proteins on their surface, as we learned last semester, and the proteins that are found on the surfaces of our own cells, our immune system ignores, but they are still antigens to somebody else. If I was to give you a kidney your body would try and reject that kidney because it would see it as foreign, because the antigens that are on it would be mine, not yours. And you would have a specific response against the antigens. So there's a self and non-self. Sometimes things can almost act like antigens, but they're too small. They're called haptens. And they don't become antigens until they combine with a larger molecule that the body makes. An example of this would be poison ivy. I've never had poison ivy because I don't make the large molecule that poison ivy needs to attach to in order to elicit an immune response against it. Cat dandruff is another one. So again, I don't make that carrier, so I'm not allergic to cats. If you make that carrier protein, you will have these antigens and you will have an immune response against it. I apologize for the noise in the background. The neighbors are obviously doing some work. So I think of haptens as half antigens. They do not have immunogenicity. They do not create immune. Antigen generates the generates the response. So the lymphocytes are made in the bone marrow. The lymphocytes are the main cells of the specific immunity. Now, there's two real groups of lymphocytes. There's three, really. There's the natural killer cells, which are an undifferentiated lymphocyte. There are the I'm going to pause this for a second. There, it looks like the motor noises have stopped. Okay, so there's a group of lymphocytes that learn the antigen that they are going to be programmed for. They get programmed in the thymus. They become immunocompetent in the thymus. And so we call them T lymphocytes. There are other lymphocytes that become immunocompetent in the bone marrow, and then we call them B lymphocytes. Now, these B and T lymphocytes are made in the bone marrow, they go into circulation in the blood, and they end up in the lymph nodes, in the spleen, and in the malt, in the linings of the guts. This is a B lymphocyte, such as it is. It looks like onion bhaji, I think. So this is just saying they come from the bone marrow, they become competent in the thymus, end up in the lymph node. These originate in the bone marrow, become competent in the bone marrow, and end up in the lymphatic system. T for thymus, B for bone marrow. Okay, any of these lymphocytes are naive. They have a receptor for a specific antigen, but if they don't encounter that antigen, they never become activated. So in order to activate, they have an encounter with the antigen. Now, T cells are going to, especially the cytotoxic T cells, the killer T cells, their job is to kill our own body cells that have a virus or a problem with them, the same as natural killer cells, except they're looking for specific problems. And because their job is to attack our own body cells that have a pathogen in them, they're going to be activated by a fragment of the antigen presented on the surface of a body cell. So to activate them, we need something called an antigen-presenting cell. Now, usually that's a macrophage that has eaten and phagocytized the pathogen and presenting the antigen to the T cell. Sometimes it can be a B cell that's been activated and will present part of the antigen to the T cell. Usually it's a phagocyte. So the macrophage will eat the pathogen and present some of the proteins from the pathogen on its surface to be an antigen-presenting cell. So when the T cell encounters the fragment of the antigen that matches their receptors, they become activated. Now, when the activated T cells will interact with the antigen-bearing cells. So it's a cell-to-cell contact, so we call this the cellular-mediated immunity. Now, T cells also synthesize these hormone-type things called cytokines, and they enhance cellular responses. There's a couple of different types of T cells. There are the helper T cells that kind of coordinate everything. There's the killer T cells that do the killing. They kill our own body cells that have something wrong with them, much like a natural killer cell. And they do it the same way, releasing perforin, which opens pores in the cell membrane. There are suppressor T cells and a couple of other different types, and there's some memory T cells. We're going to talk about these in a second. So what's going to happen here is when a T cell becomes activated, it's going to the first thing it's going to do is clone itself. It's going to make a whole bunch of copies of itself so that it basically increases the army of lymphocytes that are designed to go after this specific antigen. It just increases itself in numbers. Some of these progeny cells will go on to differentiate and become effector cells. So if it's a killer T cell, a cytotoxic T cell, they will become cytotoxic. They will start to kill. Some of them become what are called memory cells. They don't start attacking other cells. What they do is they're there for future reference, so that the second time you encounter, you have a lot of T lymphocytes that will recognize that antigen. And so the second time you encounter it, you are better prepared. These are called memory cells. Now, to use an analogy, think of the T cells as like a SWAT team. The killer T is going to do the killing. He's the sniper up on the roof, if you ever watch Flashpoint. He identifies the target, wants to attack and kill the target. But before he pulls the trigger, he radios the captain in the Winnebago and says, okay, the target's acquired, shall I take the shot? And then the guy in the Winnebago verifies that, yes, that's in fact the target. Go ahead, take the shot. And then the bad guy gets killed. You need that double check. And the T lymphocytes are the same way. And the helper T is the guy in the Winnebago. So the killer T will do the killing. It needs to be stimulated by an antigen fragment on the surface of the cell it's going to kill, but it needs to be co-stimulated by the helper T cell, because we can't have these killer T cells killing our own body cells haphazardly. We need to double check. And so the helper T cell's job is to double check. The helper T cells also, another one of their jobs is to stimulate B cells. So they're also coordinating other actions, maybe the negotiators, maybe, regular street cops, whatever. The helper T cell is the one in the Winnebago in control. The cytotoxic killer cells are the ones that actually do the killing. But before any of that happens, these cells make clones of themselves and proliferate. So the macrophage eats, displays the antigen activates the killer T cell, the antigen presenting cell activates the helper T cell, the helper T cells proliferate, the killer T cells proliferate, you get memory killer Ts, you get memory helpers, you get active killer Ts, right? You get a co-stimulation, and the killer Ts kill the memory Ts later on. Now, B cells, and we might as well do this in this thing, B cells get activated by meeting the antigen on the pathogen in the interstitial fluid, because the B cells work in the interstitial fluid. What happens is the B cell gets activated either by a helper T or by an antigen on the pathogen. When it gets stimulated, it will make copies of itself. It will clone. So this is it being activated. It will clone itself to become a whole bunch of copies of the same thing. Some of them will become memory cells, and some of the progeny become plasma cells. Basically, what they've done is they grow endoplasmic reticulum, so that's why they're plasma cells, which are protein factories. The plasma cells produce antibodies by the hundreds of thousands per hour, per minute probably. And the antibodies do what needs to be done. So the B cells become activated, produce clones. B cells need the helper cells usually, but it's not necessary. Right? The B cells differentiate into plasma cells that secrete antibodies. Antibodies, the immunoglobulins, come from plasma cells. Plasma cells are B cells that have grown endoplasmic reticulum. And then we end up with memory cells from that. So this is just the steps. So antigen-bearing agents, so the pathogens come into the tissue, the B cell encounters the antigen that fits its receptor, either alone or with the helper T cell. The B cell proliferates, making clones. Some of the clones become plasma cells, and the plasma cells synthesize the antibodies. Some of them become memory cells. With the T cells, the pathogen enters, a macrophage eats it, the macrophage get presented on the surface of the antigen-presenting cell. The helper T cell becomes activated when it encounters that displayed antigen. The activated helper T cell will activate B cells if necessary. And then antibodies are produced. Okay, so there's 5 different types of antibodies. MADGE, MADGE. IgGs are the ones that we think of. It activates complement. It's found in plasma, in your blood, in tissue fluids. It's the most common, and it's the most one of the most important ones. IgA, we secrete it, so it's in breast milk, it's in saliva, tears. So it actually helps prevent against antigens that are outside of our body. IgM is an agglutinating one. It joins pathogens together. Could be red blood cells. IgD is really the receptor on the B lymphocyte, the plasma cells, and IgE is on basophils and mast cells, and so it gets activated, and the mast cell will release inflammatory chemicals, so it's involved with inflammation. So what do antibodies do? Antibodies will stick to the antigens of whatever is going on, like of the pathogen. And so as it sticks to the outside of the pathogen, all over, it's like swarming it. It is neutralizing the pathogen by just attaching a whole bunch of antibodies to it. It also does something, they can do something called agglutination. And basically it's like a chain gang. It's like when they have prisoners get out of a paddy wagon, all their ankles are handcuffed or cuffed to a chain, so they can't get away from each other. It's harder for them all to run away coordinating. So agglutinating allows them to not get away from the pathogens. Neutralizing means they can't do anything. Precipitation means that any pathogen that's in solution comes out of solution and really becomes a precipitate. It falls out of solution and the macrophages get to eat it. So agglutination, precipitation, neutralization. It also activates complement. So when the antibody attaches to the antigen, that antibody-antigen complex on the cell membrane is where the complement goes. And that results in this opsonization, which is really making tasty, which is giving the handle for the macrophage to grab hold of. It also then affects inflammation and lysis of the pathogen, poking the hole, the membrane attack complex. So by activating the complement, it does all the things the complement does, including increasing inflammation. So that inflammation, therefore, is kind of triggered by these antibodies. So we've already talked about all the things that complement does. So when you've never encountered this pathogen before, this antigen before, then it's called the primary immune response. And so it takes quite some time for all of these steps to be activated, all this proliferation, all of this stuff to go on, before you get enough antibodies that it actually neutralizes, precipitates, agglutinates, and activates the complement against that pathogen. But the fact that we end up with memory cells, memory T cells, memory helper Ts, memory B cells, means that the second time, because they remain dormant, but they remember the antigen. They are specific for that antigen, and now you have a lot more of them. So the second time you encounter that pathogen, you ramp up this production far quicker. and you will have this response quicker, usually before the pathogen can get much of a foothold, so you don't get sick. And so this is called the secondary immune response. The secondary response is way faster and way stronger. And then on top of everything else, the antibodies that you've produced in either one primary or secondary responses, those antibodies hang around for quite some time. It might be months or it might be years. And that provides protection against that pathogen without even having to evoke another immune response. It just does it passively. So this is just a graph. The secondary response is sooner and stronger. Okay, so we can get immunity naturally. So it can be acquired by just encountering the pathogen naturally, so you get sick. And so, you know, when I was a kid, I got the chicken pox. Now I'm immune to the chicken pox. When I was a kid, I had the measles. Now I'm immune to the measles. When I was a kid, I got the mumps. Now I'm immune to the mumps because I have gone through that primary immune response, and I've made those antibodies, and I've made those memory cells, and etc. Now, another way that you can get it is artificially, and that is through vaccination. So you guys probably got the MMR, the measles, mumps, rubella vaccination, which doesn't give you the measles or the mumps or the red measles. It probably, what they did was they gave you the antigens of those things, and then you had an immune response against the antigens, not against the pathogen. And now you are protected because your primary encounter you didn't need to get sick. You're now at the secondary encounter stage. So if you actually meet those pathogens, you will have the secondary response instead of the primary response. So this is the artificial way. And both of those that I've just talked about are artificial active because you've had the response, you've made the memory cells, you've made your own antibodies, you're actively involved in the immune response. But there's a way of getting the antibodies passively. The natural way of getting antibodies passively is your mother, while you're in the womb, your mother encounters a pathogen, has an immune response against the pathogen, makes the antibodies cross the placenta, and now the baby has those antibodies circulating in their blood, so that when the baby's born, presumably that pathogen is in the environment and the baby has protection against that pathogen that the mother had the immune response to. The baby hasn't actively made their own antibodies. Mom has made the antibodies. Antibodies can cross in breast milk as well, doing the same thing. Mom's antibodies with her immune response against a pathogen can be transferred to the baby via breast milk. Both of those are natural passive immunity. The baby has not had an immune response, and therefore, if they encounter that pathogen later, it will be a primary response, not a secondary response. There's an artificial way of getting passive immunity too, and that is by an injection of something called gamma globulin. These are antibodies. So, for instance, snake bite. If there's snake venom, you'll die before you will actually have the proper immune response against the venom. So there's no time. You don't have a couple of days. So what they do is they take the venom from a cobra or whatever snake or a rattlesnake, and they give small quantities of that to a larger animal, like a horse or a cow or something, and that animal will have an immune response against it. Their plasma cells will make antibodies against that snake venom. And then we take a unit of that animal's blood, centrifuge it down, get rid of the red blood cells, get rid of the plasma, and just take the immunoglobulins from it, and we can end up with the antibodies against that snake venom. Now, there are ways in the lab that we can artificially then reproduce those antibodies, but classically it was you gave it to an animal. And it still has to start that way as far as I know. And then you get bitten by a snake, you go to the hospital, they inject you with the antibodies that they have in a serum, in a bottle, and you now have antibodies against that, and it neutralizes, agglutinates, and precipitates the snake venom, and then your macrophages eat it, and it's gone from your body. So passive, you don't make the antibodies. Active, you do make the antibodies. Natural is natural events. Artificial means that you've had an injection. So vaccination is artificial, active. Mother's milk is natural, passive. Getting sick is natural, active. And a gamma globulin serum is natural, passive. And so this is just exactly what I've just talked about. A chart saying the exact same thing. Now, the rest of these slides are all about pathologies, and we're not going to get into the pathologies at all. That's not the purpose of this course. But I would like to mention something about autoimmune disorders and autoimmunity. Autoimmunity happens when your antibodies don't recognize your self-antigens as self. And so what happens is your immune system starts attacking your own tissues, whatever they are, and you get damage from it. So things like in rheumatoid arthritis, your immune system attacks your synovial membranes. In diabetes type 1, diabetes mellitus type 1, your immune system attacks the beta cells of your pancreas. In lupus, your immune system attacks the DNA of any cells of your body that are in replication, et cetera, et cetera. These are autoantibodies. You don't want this, but it's useful to talk about.